Hello, I'm James Graham, CEO of Recce Pharmaceuticals Limited, ASX RCE. Thank you for joining today's Key Opinion Leader event, Clinical Data Milestones and the Recce Innovation Journey. Joining us today is Professor Eugene Athan, Principal Investigator of Recce's recently successful and completed Phase 2 clinical study. Professor Athan will be discussing the study and some of the groundbreaking insights of Recce 327 on a broad range of bacterial infections. Dr. Alan Dunton will be discussing the positive implications of completing this study and the Phase 3 trials just ahead of us. Dr. Sahini Saka, Principal Investigator of a anti-infective research unit at Murdoch Children's Research Institute on Recce's technologies. We hope you enjoy today's presentation.
Hi everyone, good afternoon. My name's Eugene Athan. I'm a Professor of Infectious Disease and I've been the Principal Investigator for an exciting new Phase 2 trial, the Recce product of 327, which is a topical gel which seems to have worked very effectively in the setting of acute bacterial skin and skin structure infections, and I'll be talking a bit about that trial. By way of background, I'm a professor at the School of Medicine. I'm a co-director for a research centre which looks at infectious disease and immunology research at Deakin University. And also I'm a specialist at Barwon Health, one of the largest regional health services in Australia. And I also head up the public health unit at the Barwon Southwest region under the Victorian government. So what is an acute bacterial skin and skin structure infection or diabetic infection? These are common. We see skin and soft tissue infection every day in both general practice, primary care and in hospitals. They relate to underlying risk factors, sometimes associated with trauma. In the area of diabetes, it's particularly quite a serious condition for a whole lot of reasons. People lose feeling in their feet, then they can develop trauma, ulcers, complicated infection, and can even result in potential amputation. This is one of the most important causes of disease in diabetes seen globally. And the pathogens, we know golden staph and a whole lot of other potential gram-negatives or superbugs, and in the majority of cases up to 80% are polymicrobial, so different germs, but superbugs relate to antibiotic resistance. The readmission rate for diabetic foot infections is about 40%, and there's a significant mortality at 1 year associated with that. So this is a serious disease and needs major alternative attention besides conventional approaches to treatment. We've seen increasing rates of golden staph or MRSA, methicillin-resistant Staph aureus, patients in hospital, and this is a challenging infection to treat with limited antibiotic options at the moment. So current therapy includes wound care, surgery, as I mentioned, broad spectrum antibiotics. Acute bacterial skin and skin structure infections are a significant healthcare problem, encompassing indications resulting in necrotizing fasciitis or flesh-eating bacteria, post-operative wound infections, and I've already mentioned diabetic foot infections. There's been no placebo-controlled trials because it is considered to be unethical to withhold standard treatment for patients with infection. What about the Phase 2 Recce 327 topical gel? So this is a product that seems to have extremely good efficacy against many bacteria, so very broad spectrum efficacy. And this is the first time that we've trialled it as a gel to be delivered to skin and skin structure infections or for diabetic ulcers with infection. So the Phase 2 clinical trial is an open label efficacy study, exploratory evaluation of the bioavailability of single and multi-dose R27 topical gel applied to acute bacterial skin structure infection. It's been used in both men and women, only adults, no age limit, upper age limit. The study aimed to provide critical data on the gel's effectiveness in treating a broad spectrum group of acute bacterial skin and skin structure infection, including diabetic foot infections particularly. The trial site at Barwon Health is one of the largest regional health services in Australia, and we provide access to diverse patient populations. What were the endpoints that we achieved during the trial? We achieved all primary and secondary endpoints as an open label trial, evaluating the efficacy, safety and tolerability of the drug. We also looked at pharmacokinetics. So what were the blood levels of the drug, the agent, when it was applied directly to infected wounds? We enrolled 30 patients, 29 included in the final analysis. One patient was withdrawn due to pre-existing pain at the wound site that was not related to the topical treatment. After 7 days of treatment, 86% of patients, so 25 out of 29, treated with our 327 gel had successful clinical response. At 14 days, up to 93% achieved primary efficacy endpoint. In other words, responding in terms of local inflammation or signs of infection. So we can say that R327 gel was demonstrated to be safe and well tolerated, achieving all the study endpoints with no serious adverse events reported. In particular, the study endpoints that we looked at were the Lipsky scale, which measures inflammation in diabetic ulcers or wounds, and we used wound assessment tools as well. And these are validated tools. The FDA accepted diagnosis tools, assessment severity of patient wounds. So the endpoints that I've mentioned to evaluate the efficacy of 327 topical gel in skin and skin structure infection, evaluate the potential for systemic bioavailability of single and multiple dose topical gel applied once daily for 7 or 14 days. The diabetic foot infection, we mentioned the Lipsky scale, clinical resolution of infection, and this is accepted by the FDA as a validated measure of outcome measure. 7 parameters were graded 0 to 3 for 21 total points. The higher the score, the worse the diabetic foot infection. We look for things like erythema or redness, exudate, such as purulent discharge, induration, thickening, tenderness, pain, and warmth. And then the Bates-Jensen wound assessment tool, also accepted by the FDA, shows parameters graded 0 to 5 for 65 points. Higher the score, worse the skin structure wound. We measure things like size, depth, the edge, any undermining of the tissue, any dead or necrotic tissue, any exudate, fluid around the wound, skin color, surrounding changes, swelling or edema, thickening or induration, granulation of healing, and epithelialization. So these are the methods that we use to evaluate patient wound healing and subsequently rated patients as either cured or improved, and they're validated and accepted by the FDA. So the potential for Recce 327 gel for future applications. We saw no serious adverse events during our trial. The results demonstrated the potential to address the skin and skin structure infection market. And this has been a considerable challenge, as I've already said, big burden of disease to the healthcare system. And those at high risk of skin infections and poor outcomes are often diabetic patients in particular. We saw a rapid onset of efficacy of 327. People responded mainly within the first week. And there's obviously a much more significant clinical setting for the potential of this agent to be used in different infection contexts. So plenty of therapeutic potential. And these are my references. So thank you for your attention.
Hello, I'm Dr. Alan Dunton. I'm the Chief Medical Advisor, and I'm also a non-executive director of Recce Pharmaceuticals. I've been involved intimately with the clinical program at Recce with our exciting new technology as antibacterials and antibiotics. And today I'd like to discuss with you an overview of the clinical program and our phase 3 plans going forward for Recce's 327 gel. First thing I'd like to do is to share with you our product pipeline as it stands today. Let's start at the top and work our way down. Our lead project and program right now is with our topical gel called R327 or Recce 327. And it's a very exciting time for us because we have just announced very significant positive phase 2 results, which I'll go into a little bit detail later, of patients with diabetic foot infections. It was actually a study in acute bacterial skin and soft tissue infection, which included diabetic foot infections. We will be commencing a phase 3 trial of this product in Indonesia a little bit later this year. And I'll go into that detail as well in terms of the design of the study, as well as our interactions with the Indonesian Badan, which is their Food and Drug Administration. We'll also be starting a phase 2b/3 registrational trial in Australia later this year, which is exciting. I'll also talk about our regulatory strategy later and how we are going to be moving forward with this product. Because we will get to the United States to do registration trials as well with the same product, and I'll show you how we get there. In addition to diabetic foot infection, we'll be doing studies using the topical gel 327 in acute bacterial skin and skin structure infections to do additional work on other type of wound infections. We actually started our clinical program with the intravenous formulation of Recce 327. We completed phase 1. We studied up to 4,000. We studied up to 6,000 milligrams of the drug. Up to 4,000 was extremely well tolerated as a 1-hour infusion. The drug is extremely safe. It does not change anything we've measured, including the clinical chemistries, coagulation, blood counts, electrocardiograms, telemetry. It's a very safe product. We understand the kinetics of the drug. Once you give it intravenously, it will remain in the body for 1 to 3 hours, depending on the dose. And it probably distributes fairly broadly across the entire human body. It's excreted primarily through a urine pathway, which makes it a beautiful candidate for a complicated urinary tract infection. We were able to show in urine that we collected from healthy volunteers that received the drug that urine, which contained Recce 327 in high concentrations, was able to kill E. coli, which is the most common organism one sees in urinary tract infections, and is responsible for about 1/3 of the patients who have sepsis, get it from an E. coli urinary tract infection. We'll also be looking at other types of serious infections as well as we continue development for our product, including hospital and ventilator-acquired pneumonia, which sees many resistant bugs that get formed as patients are in ICU type settings. We're also developing an aerosol for inhalation. It has not been in human trials, but it has been in animal studies and has shown very promising results to treat these same multi-drug resistant infections that are mostly hospital-acquired and through ventilators that are used on patients can be contaminated with these types of bugs and other serious lung infections. We have a very close collaboration with the United States Department of Defense, where we're studying burn wound infections. We have done a phase 1 trial using a topical spray in patients with burn wounds. Burn wound infections. It is very well tolerated, and it shows very early signs of being able to treat infections. So a burn wound is perfectly suited for a topical gel, as we're studying today. But our first trials, as I mentioned earlier, will be in diabetic foot infections. So let's move on. Let's talk about the phase 2 trial that we just announced significant results in. This was a study which was completed in 30 patients in 3 sites in Australia. It included patients with acute bacterial skin and soft tissue infections, which included diabetic foot ulcer infections as well. The primary efficacy endpoint in this study is a clinical endpoint where the physician who is the investigator in the study evaluates the wound and says, yes, the infection is cured or it's much improved. And to guide them in making that clinical assessment, they use a scale which is known as the Lipsky scale. Lipsky was a professor of medicine who developed this scale some 20 years ago, which has 7 key components where the clinician will look at, is the wound red? Is it swollen? Is it exuding pus-like liquid or clear liquid? And there's two or three other parameters there, which they rate from a scale of 0, which means it's absent, to 3, which means it's very present or very significant. So they can follow that scale in helping them determine whether or not a wound infection has been cured. Now, the very good news is that 93% of these patients that were enrolled in the study and were treated up to 14 days either cured or had significant clinical improvement. And the remarkable thing is this drug has a very fast onset of action, and some actually achieved cure within 7 days as well. They didn't have to be dosed for the entire 14-day treatment. So this is a very strong signal that there's good activity of the drug, and the scale that we used in this study will bring forward as well to use in our phase 3 study. Why do we use this scale? This is a scale which has been blessed, if you will, or approved by FDA to use in these types of clinical trials. Other drugs have been approved based on the use of that scale. And if the regulatory authorities like anything, they like a scale that's been used for drugs they have seen before. So we will use that in our registrational phase 3 study in Indonesia, which is going to start very soon. So this confirms here's a drug that's a broad spectrum antibiotic, and it's exactly what's needed to go forward to treat these kind of infections, not only for multi-drug resistant organisms, but also for infections which contain more traditional bugs, if you will, because it has such a rapid onset of effect and predictable efficacy. Moving on, let's talk a bit about the phase 3 trial that we will conduct in diabetic foot infection, or DFI, in Indonesia. So why did we go to Indonesia? Well, Indonesia actually reached out to Recce through the Australian government. Why did they do that? They wanted to make a dramatic change for their 300 million people in Indonesia to bring in the new products, the new high-tech products, which could help them cure or treat some of the persistent and significant health issues in Indonesia. About 11–12% of the people in Indonesia have diabetes, and a high percentage of those people have foot ulcers, which get infected. There's not a good solution to that problem yet, meaning a good antibiotic that will treat that. And they asked to speak to us to talk about our product, Recce 327. I was fortunate to be there in the initial discussions and working with the Biden POM, which is their FDA, or BPOM for short. It was a very cordial meeting. We described a potential protocol, which you'll see here, where the topical drug would be administered once daily for 14 days. And we would treat approximately 300 patients. 200 would be on the active product, meaning it would have 327 contained, and 100 would be randomized to placebo. Again, the primary endpoint is this clinical response I just addressed, which is the Lipsky scale, where you say, look, the wound is cured or it's dramatically improved. That's the endpoint of efficacy. And obviously, we're measuring safety as well. What we've introduced as well, and we committed to the BPOM, is that we would do an interim analysis after 100 patients have been treated. Why would we do this? Well, we would look at, is the drug continuing to be safe? And in our interim that we will conduct is we would also look at the efficacy of the drug. So we'll be continuing to have our discussions with BPOM as we move forward with this trial, which will be at no more than 10 centers across Indonesia. As I mentioned, this is a very significant medical problem in Indonesia, and we hope enrollment will go quickly. The other point I wanted to make is patients will be seen daily in their clinic where the study is ongoing in Indonesia. So if a patient is randomized to a placebo, and if that wound infection worsens or doesn't get better, the investigator can choose to drop that patient from the clinical study and put them on another antibiotic of choice. We don't want patients to have to continue for a complete 14 days with a wound infection. That would be unethical. But Biden-Palm has agreed that a placebo-controlled trial is the way to go for the first study in this registrational study in Indonesia. So that's very exciting. We're fortunate to have been introduced to them, and we look forward to our ongoing relationship. And this is a photo. I'm not in this photo. This was a subsequent visit of the team we met with at the Biden-Palm. Our CEO is in there, as well as the head of the Biden-Palm and his associates. We've been awarded by them expedited regulatory review. So it's, in essence, fast-track protocol, rolling review of the data, excuse me. And we expect with positive results from this trial, we'll be able to file an NDA in Indonesia. They're motivated to make this happen. Our phase 2 results are very, very better than I thought they would actually be. So this drug has very significant activity. So another reason for doing the study in Indonesia is that they are viewed very highly in the ASEAN region of countries, which would accept these data for the most part for approval within their individual countries. Now, ASEAN is 680 million people, right? Of those, about 300 million are from Indonesia. So it's in Southeast Asia primarily, and these countries would have very similar problems. Some have higher or lower levels of diabetes. For example, the Philippines, I know, has a very high level of diabetes and has the same significant problem as well as other countries across the region. So going ahead here, this is a very large market on a global basis, not only the ASEAN market. There's estimates out there that the diabetic foot infection market is estimated to be worth about $5.2 billion. This is US dollars. The Indonesian market in and of itself to start is probably around $200 million. And as I mentioned earlier, it's about 11% of the population that is impacted by this problem. So this is why the Indonesian regulatory authorities came to talk to us, because they had a problem and that we can help fix it. So Indonesian approvals, as I mentioned, would provide broader access to the Asian market, which is worth probably in the neighborhood of $1 billion per year. And then you can add on that, what is the Australian market, what's the US market, and it gets to be a very significant number going forward. So this is a very attractive market opportunity. What about drug resistance in general, antimicrobial resistance? And here are some insights by Dr. Nakatani out of the WHO. So far, the global environment hasn't been great in terms of tackling this issue. It's getting a lot more attention than it has. I'm sure you've seen a lot of things in the literature, including newspapers, where it's always getting worse and there's outbreaks of multi-drug resistance products. But companies haven't really gone after it and addressed it seriously. The only way microbial resistance has been addressed is by telling people to use less antibiotics. That's not the answer. The answer is to get the right antibiotic, and we think we have the correct antibiotic for use right away in patients who have infection. Why is that? That is because we don't see resistance. In a classical 30-day passage study, which is, albeit in a test tube, you culture cells, you put bacteria in there, and then you go from day to day to day where you transfer that fluid over with the cells and the bacteria to see if the bacteria become resistant. Classical antibiotics show resistance within 2 to 5 to 7 days. We've gone for 30-day passage and I've seen no signal of resistance. This is a novel technology. It's purely synthetic. There are multiple components to it. we haven't seen resistance, and we've seen no resistance in any of the trials that we've done as well. The other trials, we've repeated this passage study, and we've also now done clinical work and have no sight on any resistance. So we're really excited about it. In the broader marketplace, though, there are antibiotics that are under development, but there's only 2 new chemical classes. We're one of those new classes, and we're the furthest advanced here. So there's a lot of excitement in the scientific community, in the clinical community, and as you can see from regulatory authorities around the world and investigators. You heard from Dr. Athan, who is very well respected in this space, and he's with us on moving this product forward and is excited about it as well. So the WHO has put out a priority pathogen list, meaning which are the ones that are really critical we got to address today. You'll see at the top of the list, it's a carbapenem. A carbapenem is a class of antibiotic, a very novel and new class, but there are bugs that are resistant to carbapenems now, and that's your last stop if you're really sick and dying from it. That's the last drug you're probably going to get, or in combination with other drugs. And these bugs become resistant are Klebsiella pneumoniae and something called Acinetobacter baumannii. A lot of words, long sounding, but let me tell you, they're really nasty bugs. We have studied, and we have been fortunate to have samples from the CDC in the United States, the Centers for Disease Control, and other sources. We actually have samples of these bugs from actual clinical cases, over 500 now, and we have studied all these, albeit in a test tube, but all of them that we have that have been resistant to other antibiotics are sensitive to Recce 327. That means Recce 327 can kill these resistant species. So this is very exciting information. It's relatively recent. We actually have some clinical samples from the battlefield in the Ukraine, an unfortunate situation we all know, but these bugs, the resistant bugs that came from that battlefield, are sensitive to Recce 327. So I mentioned earlier, we'll talk about our global regulatory strategy, and it's pretty straightforward. We're working here in Australia on phase 1 and 2. We'll continue that to phase 3. We have an opportunity which we've taken to move into to Indonesia. We'll be starting our phase 3 regulatory trial very soon in Indonesia. With success there, that opens the doorway and the gateway to ASEAN countries. We'll be studying our product probably in some more clinical trials, but the Indonesian data will be fully acceptable for ASEAN countries to move forward. And those countries include Malaysia, Philippines, Singapore, and Thailand, where I know there's problems in each of those countries with diabetic foot infection. From Australia and the ASEAN countries, we have easy access to get into the United States. We will be going to the US FDA, and then subsequently we'd be going into Europe to study the Recce products as they evolve over a number of different spaces, not only the diabetic foot infection. So I appreciate your attention. I hope I was able to answer some of your questions that you may have, and hope you have a good and a successful day. Thank you very much.
Hello, I'm Dr. Shohini Sarkar. I'm a senior research fellow and a principal investigator at the Anti-Infective Research Unit set up by Recce at the Murdoch Children's Research Institute in Melbourne, Australia. I also am a part of the infection immunity and global health team over here, focusing on respiratory infections. Now, I've been working with Recce from 2021, so it's been a few years. And it's a real pleasure to present some of the interesting updates that we have from our lung studies that we have done with Recce's lead candidate at the Anti-Infective Research Unit. Now, Recce really is at the forefront of combating antimicrobial resistance. And today I'll be focusing on non-tuberculous mycobacteria. Now, most people would have heard of tuberculosis, but NTM comprise mycobacterial species other than those causing tuberculosis and leprosy. These are very closely related to MTB, But NTM are primarily opportunistic pathogens that are increasing in global prevalence. And what's of particular concern is that NTMs are particularly increasing in conflict zones and are getting more and more prevalent in infected combat-related wounds. Now, NTMs cause a wide spectrum of disease, ranging from localized skin and soft tissue infections to TB-like lung disease, which can also then become systemic or bloodstream infections. High-risk population groups include people with chronic lung disease, such as emphysema, asthma, COPD, bronchiectasis, and cystic fibrosis. Other groups also include those with immunosuppression, such as people undergoing cancer chemotherapy, people living with HIV, people with a long history of steroid use, those with kidney failure, as well as transplant recipients. And NTMs are also increasing in elderly adults. The problem with NTM infections is that they're usually highly drug resistant. Treatment failure rates are quite high compared to other bacterial infections and can be more than 50% for some NTM species. We're focusing particularly on Mycobacterium abscessus today. This is a cystic fibrosis superbug. Cystic fibrosis is one of the most common and serious genetic conditions, and it's an inherited disorder arising from a defect in the CFTR gene, which encodes for a chloride channel. It's a multi-organ disease affecting primarily the lungs and various organs of the digestive system. In the lungs, due to a buildup of thick, sticky mucus, people with CF end up getting repeated infections, resulting in inflammation, and the repeated cycle of this infection and inflammation leads to lung damage. M. abscessus is an important CF pathogen affecting almost 3 to 20% of people with CF. These infections can be extremely difficult to treat and can further accelerate the progression of lung function decline. Current treatment plans involve cocktails of various antibiotics. Treatment can typically range from a few months, can last up to 1 to 2 years, and these are also associated with high toxicities and high treatment failure rates, like I mentioned, up to 50% in some cases. Now, macrophages and these beautiful cells that you see over here, they're immune cells. They function like the vacuum cells of the body. They're part of our innate immune system. Their primary job is to recognize foreign particles, such as invading pathogens, and gobble them up, and then destroy those internally. Macrophages are a primary target of mycobacterial infections. What these mycobacteria do is once they're taken up by the macrophages, instead of getting killed in there, they're able to divide quite rapidly therein. And they're also shielded from the action of other antimicrobials while they're inside the macrophages. And then they can lyse the macrophages open and then go on to infect other cells. So they're primary intercellular source of infection, an important reservoir that can seed further infection and also lead to recurrence or treatment failure. With the infection model that we have set up, it's a human macrophage infection model with intracellular M. abscessus. And what we have seen that when we treat these infected macrophages with R327 with just 24 hours after treatment, we see a massive decrease in intracellular infection burden. This is more than a 5-log difference, which is more than 99.999% clearance of intracellular bacteria. And this effect is further made further significant after 3 days of treatment. You can see here, after 3 days of treatment, bacteria is really cleared below the level of detection. We have compared R327 to one of the clinically used antibiotics, clarithromycin, and you can see here that R327 significantly outperforms clarithromycin with respect to intracellular bacterial clearance. And what was also really pleasing to see was was that we could not detect any host cell cytotoxicity. This is really an important thing to look at, as we know that current NTM treatments, they're all associated with significant side effects. So that is really something we wish to avoid going forward. We also have a mouse lung infection model. Now, because these are opportunistic pathogens, NTM are present everywhere in the environment, ranging from air, water, soil. So most of us are exposed to NTM on a daily basis, but it's really people with those underlying complications that I detailed in the high-risk groups. They are more susceptible to NTM infections. So if we infect wild-type mice, they are rapidly cleared from wild-type mice. So there needs to be this background of some form of immune deficiency if we are to have a valid NTM infection model. So we've gone with TNF-alpha deficient mouse model. So TNF-alpha is a cytokine. It's made by immune system, and this particular pathway is also dysfunctional in people with cystic fibrosis and with other forms of immunosuppression. So with this TNF-alpha mice, when we infect them intranasally with GFP, which is a green fluorescent labeled version of the M. abscessus strain, we infect the mice. It's then able to colonize the lungs and stay there. Because we are able to establish a chronic infection, we are then also able to test a treatment protocol. In this case, we looked at 7 days of twice daily dosing via a nebulizer. And then we assess infection burden in the lungs as well as other markers on day 11 post-infection. So R327 delivery as a nebulized mist to the lung was really well tolerated. We saw no adverse effects in mouse lung infection. This is really important because not all antibiotics can be nebulized. And then again, they're not all well tolerated when you deliver them directly to the lungs. So it was really great to see R327 being well tolerated in the mouse lungs throughout the treatment period. We did not see any other side effects from routine monitoring over that 7-day time course of treatment. And you can see the mouse body weights over here tracking over time. It's no different to our untreated controls that were just given water or saline, or with the placebo control, or even the intranasal azithromycin, which was a clinical comparator that we used in this group. You may have seen this data before. It was publicized last year, but I'm just bringing it up again just to highlight the amazing efficacy that we saw from nebulized R327 treatment. Again, I do have to mention that azithromycin could not be delivered as a nebulized mist due to solubility issues. This is a major problem with a lot of the antibiotics, while nebulization has greater penetration into the lung tissue, which a lot of the antibiotics cannot effectively reach. Not all antibiotics can be solubilized, and even then they can't be nebulized, or they may not have good safety profile in the nebulized formulation. So that was the best control that we could have in this particular model at the time. And with our 327 treatment, we can see more than 99.9% clearance of the lung bacterial load after that 7-day treatment protocol. I would also like to mention that M. abscessus, like other NTM species, can cause skin infections as well. In fact, it was first isolated from a skin abscess, hence the name abscessus. You can see here on the right a keratinocyte cell line. It's an important and one of the major cell groups of the uppermost layer of our skin. And in this particular assay, we grow the skin cells in a culture dish, which we are then able to infect with M. abscessus. And the way this assay is performed, we can look at extracellular or adherent bacteria, and we can also look at bacteria that have invaded the keratinocyte or the skin cell and gone inside and look at the bacterial burden in both of these extra and intracellular regions. So in our infection model, we compared R327 with not only a placebo control, but also clarithromycin, which is an antibiotic used in the clinic. You can see here with R327, just an hour after treatment, there is more than 4 log reduction in bacterial burden. And you can see this reduction is also greater than that offered by clarithromycin. And what was really pleasing to see was that this effect was retained even after 3 days of treatment, whereas you can see the numbers treated with clarithromycin slowly going up. Now, moving on to the invasive or intracellular compartment, you can see here again a similar effect where we see a quite drastic reduction an hour post-treatment, which is then retained up to 3 days after treatment, whereas the clinical comparator there is again creeping up slowly. So overall, this shows that R327 is effective in treating extracellular as well as intracellular infection. So in summary, I would like to highlight R327 as a promising candidate for treating NTM infections. Now NTM and the diverse species belonging to this particular group of mycobacteria, they can cause a wide range of infections, like I've shown before, ranging from skin and soft tissue to TB-like lung disease. And I hope we have been able to show you that R327 shows promising efficacy against both lung and skin infections caused by M. abscessus, which is one of the most common and highly resistant NTM species. And our lung study data now demonstrates impact in respiratory function, with showing great efficacy and providing new hope for addressing anti-infective challenges in dealing with these problematic group of pathogens. Nebulized R327 can be delivered safely in our model while retaining its efficacy. That's really important to highlight as well. And while current NTM therapeutics are associated with long treatment times and significant adverse effects and high treatment failure rates, R327 presents a viable alternative as a safe and effective treatment option that will improve patient outcomes. Thank you for your time today and listening to my presentation. We are really excited by some of the data that is coming through our anti-infective research unit in Melbourne, and I look forward to presenting some more updates in the future. Stay tuned.
Thank you, Sahini. I'll now provide a quick company update on Recce's activities. So as an overview, Recce Pharmaceuticals is listed on the Australian Stock Exchange under ticker code RCE. We are an Australian clinical stage biotech with a United States presence and developing the first new class of anti-infectives in over 40 years. A class that has a unique mechanism of action that allows it to be capable against a broad spectrum of infections, particularly those of serious and life-threatening indications. The technology is founded by 40 granted patents all around the world out to 2041. As you have seen, we have completed a successful phase 2 clinical study in Australia, and we have a registrational phase 3 study, in fact, in Indonesia. We will be bringing on board an additional phase 3 study in Australia that really allows us to have the opportunity of the ASEAN group of countries through Indonesia, as well as the study of Australia for the Western medicine side of things. Working with the US Department of Defense, we have a United States congressionally directed research program that's focused upon the military burn wounds. It's a program supported by around $3 million Australian grant from the US government, and we look forward to updating upon its milestones. Our goal is regulatory approval and commercialization. And we really see that with our Indonesian study starting imminently, of course, across Indonesia for entry into the ASEAN, that study can conclude within this calendar year for sales into 2026. What is special about us and why are we different? Well, the World Health Organization, in fact, lists us as the first new class in over 40 years, the one of a particularly novel mechanism of action, and the most capable against the suite of escape pathogens that are the deadliest escape pathogens faced in antibiotic resistance to date. The US FDA maintains that we have the only qualified infectious disease designation, which is a legal designation, providing an additional 10 years of market exclusivity post-approval, plus fast-track regulatory review for the indication of bacteremia, which is the technical term for broad-spectrum sepsis. So why are we different? We are unprecedented in our broad-spectrum activity. We have been tested in over 400 clinical strains, including some clinical strains from the front line of the Ukraine battlefield, in fact, and have worked against all, and importantly, keep on working with repeated use. Why? It is driven by our novel universal mechanism of action. We have a rapid onset of effect measured in minutes as compared to traditional hours of existing antibiotics. We are entirely synthetic. All antibiotics to date are naturally derived. They're bacterial, fungi, cultivated out, and you're really as good as what's found in nature. We began with first principles in mind and created a polymeric compound that's able to work and keep on working with repeated use. Be manufactured with a very low cost of raw materials with 100% yield. Be soluble at all pHs of the blood to the opposite acidity of the stomach, and work and keep on working with repeated use, which means you have a product that is valuable today and valuable into the future. The bacteria will always mutate, but they will never overcome the dozens and dozens of mechanisms of action built into this compound. We have an ability to entirely avoid the healthy eukaryotic cell and be attracted to, bind to, and interact with the deadly bacterial cell, because there's no such thing as good bacteria in your blood or your wounds, as we see through these presentations, and that's really where we're most effective. We're on track to be the only clinical stage company, but in a commercial setting, that's capable against the full suite of escape pathogens. Here, we talk about granted patents. Why? Because a company in the pharmaceutical space is particularly founded upon the technology and market exclusivities that are granted to it. This is over 80% of the world's pharmaceutical markets. Our family one is our particularly focused upon our method of manufacture. Our family two builds upon that method of manufacture, extending the claims out and including methods of administration against human bacterial infections. Family 3 is broadening out the methods of treatments to include viral infections. And Family 4 reaffirms and renews our manufacturing claims and broadens our antibacterial, antiviral, and even some anti-cancer polymeric capabilities of our compounds. And we see that granted status taking us out to 2041. So a long, bright market monopoly future ahead. What does that really position us as? An incredibly unique, in fact, entirely unprecedented, novel, synthetic, broad-spectrum anti-infective. We have demonstrated an incredibly successful, in fact, groundbreaking phase 2 clinical study that allows us to have a dual-pronged commercial strategy into Indonesia, which, if approved in Indonesia, is approved into the ASEAN group of countries, likely then across into the Middle East, South Africa, and potentially the UK, whilst the Australia continues the FDA strategy focused upon the Americas. Our market monopolies are supported by over 40 granted patents. Our manufacturing is producing to both quality and volume to phase 3 and commercial scale, early commercial scale, I must say, for our time ahead. We are the only World Health Organization approved compound for ATP synthesis, or the disruptive factor against the ATP, or the beating heart of bacterial cells. That's an incredibly unique mechanism of action. It is the first new class of antibiotic or anti-infective in over 40 years. It is recognized by the US FDA under a qualified infectious disease designation. We are sponsored within the United States Department of Defense, Army, as a burn wound agent. And we really look forward to updating upon the clinical successes that we have had and those into the future. And we thank you for your interest and joining us on this journey.